Rapid Pyrase Test for Bacterial Identification. Learn how to perform the Pyrase test, a quick and easy method for identifying group A Strep and Enterococci. This video covers the principle behind the test, step-by-step instructions, and interpretation of results. Don't forget to subscribe for more microbiology tutorials. General information on the Pyrase test. The pyrrolidinylaralamidase, abbreviation PYR, test is a rapid method used for the presumptive identification of group A beta hemolytic streptococci and enterococci. This test relies on the ability of these bacteria to produce the enzyme Pyrase, which hydrolyzes the substrate L-naphthylamide sharp S-naphthylamide. The hydrolysis releases free sharp S-naphthylamide, which is then detected by adding N-dimethylaminosinamaldehyde. This reagent reacts with the sharp S-naphthylamide to produce a red Schiff base, indicating a positive result. Performing the Pyrase test. The PYR test is often performed using a slide, which contains dried chromogenic substrates for both Pyrase and beta-gluconidase. To perform the test, a few colonies of the bacteria in question are applied to the test strip. After a 2-minute incubation period, the results are interpreted. A positive result is indicated by the development of a red color, confirming the presence of Pyrase activity in S. pyogenes, E. fecalis, and E. faecium. A negative result is indicated by no color change or a blue color, which may be due to a positive indole reaction. Bacteria that typically yield a negative result include group B streptococci, Streptococcus agalactiae, Streptococcus mitis, S. bovis, S. equinus, and S. milleri. It is important to note that a pale pink reaction is considered a weak result and should be interpreted as negative. O to the PYR test. A crimson hue, a telltale sign, Pyrase enzyme, oh so fine. Group A Strep, Enterococci 2, their presence revealed, in shades of red, it's true. L-naphthylamide, the substrate's call. Hydrolyzed with grace, it starts to fall. Beta-naphthylamide, now set free, reacts with reagent, for all to see. A Schiff base forms, a vibrant art, confirming the bacteria, right from the start. So hail the PYR test, swift and keen, a microbiologist's helpful scene.